Hi, my name is Angela Belcher, and I'm a professor in biological engineering at MIT. I'm also a material science engineering and part of the Cancer Institute. I'm excited to speak to you during via recording today, and then we'll have a chance for a question and answer period. So Eric said that less is more, which is a concept I really don't understand. To me, it's more is more. And so I'm going to give you kind of a walkthrough in things that I love and things that I'm fascinated at the interface between living and non-living systems, as well as biological interfaces. And so I'm going to take you through kind of a rapid, in-depth talk that's more like a scientific talk that I would give at the graduate or post graduate level. But I hope that you find it exciting, and then we can jump into a lot more of the details when we meet in person. And so I'm going to share my screen and walk through some slides. And I usually like to think about starting at the beginning of Earth and going to the beginning of Earth to what happened yesterday. Because we only have an hour or so, I might cut a little bit of that out. But pretty much since I was in middle school, I was interested in the origin of life. How did we go from these relatively unsophisticated building blocks of organic molecules that might have been in the oceans or in water to complex cells to complex beings, whether it's my cat, Himalaya, named after the Himalayas. This is Mount Everest behind me. My family and I were there in the fall, to complex organisms like us today. And you're teachers, and so you know a lot about complex organisms in your classroom. The young scientists and engineers that make up your classroom. So, and I love teaching, and I love teaching most of all because I like young people, but I also this is what I said to my own kids. I said, I hope that you meet people that love science and engineering as much as me, but you'll never meet anyone who loves it more, because I'm truly fascinated with the world around me, wanting to know how the pieces work, how they fit together, and how to build new things. And so I always say I have the best job in the world because I get to work on creating new structures, new materials, and trying to address some of the problems that I think are the most exciting problems, but also ones that affect society. And so that's pretty much what I do. So I'll walk you through that today. So let me start by sharing my screen. And so here we go. I'm gonna move. I'm not sure if I'm going to show up on this or not, but okay. So I call this giving new life to materials, because my interest since I was in undergraduate school has been at the living, non-living interface, like shells, like abalone shells, which is what I did my PhD on. And my interest is also thinking about how amazing living structures are in terms of they can reproduce, they can self-correct, and they have healing properties. Well, how do you take things like that, and how do you translate it to structures, materials in the world that haven't had the opportunity to have these wonderful living properties? And so this goes to the idea of how do you give genetic information to non-living structures? And so I'm a professor. I don't do a lot of the real work myself. The real work comes from my team, my graduate students, my postdocs, my undergraduate students. And so I'll try to think them as I go along today. In the bottom here, you can see two of my best favorite collaborators, Professor Paula Hammond, who's head of chemical engineering at MIT, and Professor Sangeeta Bhatia, who's in electrical engineering at MIT, and in the IMES, the Medical Institute on campus. And this picture here is a petri dish with the idea of, can you start to grow your own batteries? This picture up here is, can you genetically code a better solar cell? So we'll walk through that today. I'm really an engineer. I'm trained as a scientist. My PhD, well, my undergraduate degree is in creative studies, design your own major. I was interested in the origin of life, so I did it in biology, chemistry, physics, geology, the interface of those disciplines. And my PhD is in solid state chemistry, but I really consider myself an engineer because I'm very interested in problem solving. And so I've always said there's never been a better time to be a scientist or engineer than now, because we've never had more of an opportunity to make an impact in the world. And I've said that for the last 5 years. And here we are in the middle of COVID. And there's never been a more important time to be a scientist or engineer now, where we have to draw on our basic knowledge and basic science of so many disciplines in order to try to solve problems in medicine and diagnostics, and therapeutics, in this course, in this time, to make an impact on a global pandemic. So as educators, many of you as science and engineering disciplines yourself, you think about solving problems, but at the same time, being an educator, being a teacher, the role of helping to spark the imagination of our youngest to help solve the most pressing issues on the planet, I think is absolutely key. So here's some things I think are pressing: inexpensive healthcare for all, clean energy, water, and food. So these are all things that I'm interested in personally. My inspiration comes from nature, because nature has been evolving. Biology has been evolving for several billions of years to be able to solve their own problems. The problem here is this abalone shell, which is an amazing marine organism. It's a snail that makes its own calcium carbonate based shell, which a lot of my research is based on. And then here's the idea of really strong, tough structures that make up plants that are photosynthetic, that can use energy, use the sun, capture light to make their own energy sources. So to me, those are just two amazing examples of how organisms have evolved through time to have really great properties. And so when I think about future research in my lab, I think about how to give genetic information to non-living structures. And the example here is the abalone. When a male and female abalone get together, they pass on the genetic information to their offsprings that say, this is how to make an exquisite shell. The shell is 3,000 times tougher than its geological counterpart of calcium carbonate. It gives their offspring genetic information, says, go out and make your own. Same with diatoms, which are made out of silica. They're beautiful glass-based structure. They basically make glass at ocean temperatures and ocean pressures. And there's at least 10,000 different of species of diatoms. That's a scanning electron micrograph. Well, it's a colored scanning electron micrograph image of these beautiful glass structures made by nature. They have the DNA sequence that codes the protein sequence, and the glycosylation allows them to grow these beautiful materials. So when I think about future technology, I start with about 4.5 billion years ago. You think it took about 1 billion years to have life on Earth. And then really exciting things started happening. You went from very small molecules to much more complex molecules, which would become DNA and RNA and proteins. You had organisms have become from single cells to multicellularity. You had photosynthesis happening. And so all these really amazing things were happening along this geologic time period, but it wasn't until about 500 million years ago that organisms started making hard materials. Up until this point, up into the Cambrian geologic time period, things were pretty much soft and squishy. And then all of a sudden, under a pretty rapid time scale, we started to have mineralization. You had organisms that would make calcium carbonate, organisms that started making silica, organisms that started making iron phosphate based materials and calcium phosphate materials. Sorry, calcium phosphate and iron oxide based materials. So the idea is, wow, this happened over a relatively short period of time. Well, why did it happen? Well, organisms had the opportunity to start making hard materials. Organisms were soft and squishy, at this time there was increased calcium and other ions in the ocean, and organisms had to learn how to deal with it. And so they learned how to deal with it by actually taking proteins they already had, repurposing them, and using them to mineralize structures. So the basic idea of what my research is can you give organisms the ability to not code calcium carbonate or calcium phosphate or iron oxide or silica, but to basically grow a battery or grow a solar cell or grow a new material that could be used for medical diagnostics. So the idea is that you would take a simple organism like a bacteria or a virus, a bacteriophage, and reprogram it to have a new function. The bacteriophage, which is a virus that infects only bacteria, its new function would be to not infect bacteria, but to grow a battery and reproduce. So people said this idea was very crazy, called me insane and such. But it's the focus of what my research has been on. So what we really focus on in my group is the interface between nanotechnology and biomaterials. And we basically use that for applications. I'm an engineer, I only want to make useful things. And so we use it for applications in healthcare, energy, and the environment. And the whole idea is if you can start controlling materials in a very exquisite way, the same way that I showed you with those beautiful sculpted diatoms. Now, if you could do that with any kind of material you wanted, you could have a lot of applications. And so I've never been accused of focusing. And so we work on many things from energy, solar cells, batteries, fuel, CO2 sequestration storage, more abundant materials, cleaner mining, water purifications, imaging and therapeutics. And so basically, can you give biology the ability to engineer using genetics for those kinds of materials? These are some companies that have spun off in this area. And a lot of the secret to why biology can do such a good job with materials is the fact that it's nanoscale. So we all know nano. We know that nano is used in so many technologies right now, from our personal electronics to video games to cosmetics to medical devices. But the thing about nano that's really interesting is not that nano is small. I mean, it's great that things are small, but what's really important about being on the nanoscale is basically a billionth of a meter. Being on this length scale is that properties change. When you take a large bulk material, and you start to whittle it down towards just a collection of atoms, the way those atoms and the electrons in those atoms are interacting with each other changes, and it changes its properties. It changes its optical properties, it changes the mechanical properties, it changes the electrical properties, it changes the magnetic properties, and that's what we're interested in. We're interested in tuning new properties of materials at the nanoscale so we can have better device performance. So some of the things we're going to talk about today are DNA, and this is the size of DNA, which is 2.5 nanometers in diameter. It's about 1,000 times smaller than a bacteria, which is about 2.5 microns in scale, and it's about 1,000 times smaller than a raindrop. We're going to talk about single-walled carbon nanotubes, which are one of my favorite materials, one of my favorite nanomaterials, and they're about a nanometer in diameter, and they're about 100,000 times smaller than a human hair, and this is comparing the human hair to a house. And we're going to talk about nanoparticles, and a lot of the nanoparticles that we make in my lab are about 4 nanometers, actually between 4 and 20 nanometers in size, and we're going to be making them in a lot of the devices that I'm going to talk about today, and they're about 1 million times smaller than an ant. And so size matters, but to me, size matters because it changes the properties of the materials. And so this is actually just looking at some biological molecules and structures within the cell, within the human body, and how big they are relative to 100 nanometers. And so we have beta-pleated sheets and globular proteins, structures of proteins. We have ribosomes, which are the machinery of the cell that make proteins. We have antibodies, which heard a lot about recently in the news with COVID related testing, and then DNA. So they're all much smaller than 100 nanometers. And that's one thing that's so amazing about biology. One of the reasons I love biology is that biology already gives you structures in the nanoscale, and they give you perfect, beautiful structures in the nanoscale. They have a DNA sequence. The DNA sequence will code, could code for one of these proteins, the antibodies, the globular proteins or so. And you take that primary sequence of that protein and it folds into a beautiful nanostructured material. And for the most part, it folds in that beautiful nanostructured material the same every time. And so it's like, wow, we have a DNA sequence that codes for a beautiful piece of material that can go on and do something else that we're interested in doing. That's one of the reasons I love biology. Here's some examples of natural biomaterials that biology already uses and makes inorganic materials. So I'm a solid state inorganic chemist by training, so I'm really interested in inorganic materials. So this is a coccolithophorid. It's a unicellular algae made out of calcium carbonate. This is the abalone shell, which is really my favorite biomaterial next to my two kids. And if you take that calcium carbonate material, which is actually 3,000 times tougher than its geological counterpart, it's made in ocean temperatures, ocean pressures. It doesn't use toxic materials, and it doesn't add toxic materials back into the environment. There's a slug, it's not really a slug, there's a snail that makes this really exquisite, beautiful material. And it does it pretty much perfectly at the nanoscale. Now, if you take that and you hit it with a hammer and you look at it in the scanning electron micrograph, that picture next to it, what you'll see is that calcium carbonate sheets are actually made of little tablets. You can think of them as like brick wall structures that are all intercalated within each other. So that calcium carbonate material is inorganic material, but between every single layer of these tablets is an organic matrix. And so organic matrix is made of mostly proteins. These proteins are made out of the 20 different amino acids. And the proteins that are made by that snail, that marine gastropod, that abalone, grab onto the calcium out of the environment and start building this really exquisite structure every single time. Diatoms made out of silica that I talked about before, and magnetotactic bacteria, which make single domain magnets of iron oxide, Fe2O3, Fe3O4, beautiful magnets made by biology. All of these are made at ocean temperatures. There are no extreme temperatures, no extreme pressures. They're all made within the cell, within these organisms, and coded at the DNA level. Wow, life has done a really amazing job. If this is actually going in and looking at the scanning electron micrograph, looking at higher magnification, here you can see this beautiful brick wall-like structure. The individual tablets, they're pseudohexagons made out of calcium carbonate. In between every layer of that calcium carbonate is this organic matrix that sets the crystal structure, sets the crystal spacing of these materials. And these are actually kind of the ends that are growing out over time. And they also have these really special proteins that are like ligament-like structures that make them such a strong and tough structure. So all of this is, built by an organism, and pretty rapidly as well. This is actually an abalone here. I worked in the black pearl industry. Pearls, if you have pearls at home, they're made in the same way. Pearls are made out of calcium carbonate. So it's not like a really precious material. The beauty, the luster, the color of it comes from the perfection in the nanostructure material. The color comes from two different places. It comes from the thickness of the tablets, which are on the thickness is on the order of the wavelength of visible light. And it also comes from some pigments that are placed in the growing structure as well. So I love the periodic table, and one of the things a lot of things fascinate me about the periodic table, but one of the things that is really interesting to me, if you think about if life came from the ocean, it had to start to evolve with the ions that were available free in the ocean. That's why I talked about when you had increased calcium, you had increased silicon, you had increased iron. This was actually toxic to the organisms that lived in the ocean, and they had to learn how to deal with it. And they learned how to deal with it by building structures and pushing it outwards. And so the organisms that evolved in the ocean evolved with only the elements they had available at the time. So it's very high in calcium and calcium carbonate, calcium phosphate, the things I talked about before. But if you look in this part of the periodic table here, and in the transition metals, most of the periodic table was not freely available for organisms in the ocean to start working with. And so I think life did a pretty good job, but I think life would have done better, would have had more opportunity if it had more elements to work with. And so that's what I do. I try to give organisms the opportunity to expand the elements it can work with so it can make other kinds of structures that may be more technologically important to us. So pearls are beautiful, and they can be valuable. The abalone shell is definitely valuable to the abalone. The exquisite, beautiful silica-based materials are also really important to the abalone, but not that useful to me. I like them to expand what they can work with. And so I have this periodic table made for students at MIT, and I'll have one to share. I'll actually share quite a few for each of you. On one side, it says, "Welcome to MIT, now you're in your element." Because when I got to MIT, I finally realized that I really fit in. And the other side are the amino acids with their pKa's. And so, because I'm always thinking about the interface between elements and protein structures, biological structures, and so we'll share that with you. When I was pretty lucky to have President Obama visit my lab, and he was really quite fun to show around. And I gave him this periodic table when he visited, because I thought, everyone needs a periodic table. And I said, "Sir, I want to give you this periodic table in case you're ever in bind and need to calculate molecular weight." And he looked at it, he said, "Thank you, I'll look at it periodically." And then later he gave a lecture and showed, MIT, they give out, periodic tables. I guess he thought that was interesting, but it was actually a lot of fun. Also, I'll share quite a bit with all of you today. You can keep some and hand them out to your students. And so, but so I probably have convinced you that biology does a great job of making hard materials, inorganic materials, but how does it do it? Well, it does it by taking mostly proteins that they repurpose, and this time they're repurposing to make a shell. And so up here you have this picture. It says organic matrix. In this picture that says organic matrix, you can think of that as a string of amino acids. In the case of an abalone shell, it's a protein that's very negatively charged, anionic. So I'll give you a second to think about your favorite negative amino acid. Okay, so mine actually is aspartic acid. So aspartic acid and glutamic acid, these are the proteins that have a negative charge. You can think of them as that being able to grab the calcium out of the environment and build up layers of calcium carbonate. So you can think of having a protein matrix secreted by a cell that can allow you to nucleate a material of choice. And so this is how it works actually with quite a few kinds of biomaterials, including calcium phosphate-based materials that are bones. But what my lab did, what I did was try to say, well, what if I wanted biology to not work with things that it had worked with through time, but evolve the ability to work with a semiconductor material, say for the electronics industry? How would you do that? And so I wanted to be able to have a protein sequence that's specifically bound to the chemical and geometric structure of any material I was interested in, could make an imprint of it, and then grow that material and control the material on the nanoscale so we could control the properties. Because that abalone shell, there are two secrets to it. One was that it's nanostructured, and that's part of where its incredible strength comes from. And the other is evolution. It basically had the chance to evolve to be this really amazing structure. So these organisms started making these materials about 500 million years ago, but it took about 50 million years to get good at it. So not something you would do, on the time scale of a career, really. And so what my challenge was is how do you do that? How do you get organisms to increase their toolkit for inorganic materials? And so I decided to borrow an idea from the drug discovery industry. I decided to use a bacteriophage. Now, a bacteriophage is a virus that infects bacteria. It's not infectious to humans. It's harmless to humans. And the reason I decided to use that were a couple of reasons. One, it's a very, very simple structure. It has single-stranded DNA, and the single-stranded DNA code for the proteins that makes it up. And you can easily go in with enzymes, cut the DNA, add new pieces of DNA in, and make a library of viruses. So it's actually not that difficult to go in and make these, puts this picture here shows gene 3, add gene 3 here, and gene 3 codes for protein 3, so it will give you these proteins just on the tip here. Now you could go in and do that with a billion different combinations and make a billion different viruses that are almost the same, but they may differ based on 12 amino acid sequence on their tip. So why would you do that? What you want to do is you want to do what's called combinatorial chemistry, combinatorial biology, have a library. Just like you go into a library and you have, you can have thousands to millions of volumes of books. I want to have a billion different viruses that only differ in a small difference. And I want to mine those for the ability to grow a better battery, ability to grow a better solar cell material. That's the idea. So this virus is so beautiful. This bacteriophage is so beautiful. And one of the things that's so beautiful about it is it's 880 nanometers in length. So it's almost a micron in length, but it's just about 6 nanometers in diameter. And so it scales both the micron and the nanoscale at the same time, which to me is a beautiful combination. And it has only a couple of genes, and these couple of genes code for the proteins that make it up. So you can see here, this gene 3 codes for protein 3 on this end. Gene 8 codes for 2,700 copies of a protein that make up this virus. And gene 9 is over here, and it codes for proteins on this end. So I look at this as a beautiful little molecular device that I can go in and change the DNA sequences. By changing the DNA sequences, I can change the protein sequences one at a time, multiple at a time, to get them to do something for me that I want them to do. And this is done through a library approach. In this case, you have DNA. You go in and you cut and paste random DNA sequences in. These random DNA sequences code for random protein sequences. And now I can say, I know the sequence of proteins in an abalone shell that make calcium carbonate. I know some of the sequences that make calcium phosphate. I know some of the sequences that are involved in silica formation, but I have no idea what sequence I would use if I wanted to build a better solar cell or better battery. Instead of me going through and designing this, I'm going to do a combinatorial approach. I'm going to take a billion possibilities, and I'm going to force those billion possibilities to interact with my solar cell or my battery and say, which of you can imprint the surface and grow the material that I want to grow? I'm moving this around a little bit. I don't know if my picture is showing up on here or not, and this is my first time to record a lecture by Zoom. But here's the basic idea. So you take a library of 1 billion different viruses that I talked about before. That are these small differences. Now, and this billion different viruses will be only in less than a drop of liquid buffer. And you force it to interact with, say, a semiconductor material, electronic material, a battery material, a solar cell, a magnet, whatever you're interested in. And what you do is you allow them to sit on the surface, you allow them to feel the surface, chemically feel the surface, structurally feel the surface, and try to find the 1 out of 1 billion that'll recognize that surface in a way that other ones won't. So you put a bunch of them on, most of them won't stick. You look for the one that binds the best. You take that one, you infect it into its bacterial host. Now you have a bacteria, which is a natural host of this virus, make millions and billions of copies of that virus that recognized your, let's say, battery material. And now you have a collection of viruses that had some affinity for that material. But you know what? Maybe it wasn't great. You use kind of a Darwinian process. You say, okay, you did a pretty good job the first time. Now we're going to ask you to bind again under more stringent conditions, go through the process again. And now we're going to ask you to bind again. Now we're going to ask you to bind again till you're looking for one that is really standout super. Here, let's see if this works. This is kind of a movie that some of my students made a long time ago. You throw all these possibilities. We're looking for the one that has that chemical specificity. Most of them don't. They get washed away by non-specific washing. Then you remove them from the surface either by lowering the pH or through ion competition. They come off the surface. Now you need to make copies of them, so you infect them into a bacterial host, make 1 million copies. There it goes, it gets infected, it's replicated, it's given back off again, and now you have a sequence that recognizes that semiconductor that you are interested in. This is actually just showing viruses or structures are absolutely beautiful. This one I work on called M13 bacteriophage is no exception. If you're a big protein fan like me out there, it has these really beautiful alpha helixes. There's 2,700 copies of them. They're positively charged on the inside. They bind to the DNA sequence, the single-stranded DNA inside of the virus, and they self-assemble into these perfectly beautiful structures that are almost a micron long. It's 6 nanometers in diameter, and now they're perfect. You can make them the same every single time. You clone them, basically. And so now I think of the amino acids on these proteins as beautiful reaction vessels, where the amino acids differ from each other based on their chemical functionality. Are they hydrophobic? Are they hydrophilic? Do they hydrogen bond? Do they form disulfide bonds? Are they positive? Are they negative? And it's those sequences of the amino acids along these beautiful alpha helixes that are going to sequence, that are going to grab onto ions, that are going to grow the materials that we're interested in. So what am I interested in? Well, pretty much everything. This is what my students think I look like. This is what they think one of my very best collaborators looks like, Professor Paula Hammond. And the idea is we're going to give DNA sequences to come up with new routes to grow solar cells, batteries, catalysts, drop-in fuel replacements, basically build houses, artificial photosynthesis, and cancer diagnostics and imaging. These are all things that we've worked on in the past and shown that we can actually do. So why use biology to begin with? Well, one of the reasons is that it has the ability to evolve, which is important to me. But if you think about it, if you think about that abalone, or you think about that diatom or that magnetobacteria, it did so without using any toxic materials. It used no toxic materials and added no toxic materials back into the environment, which is very different on how we make batteries, how we make solar cells, and how we make electronics. And so we think it's a great model for thinking about how to make materials that are compatible with the environment in which we live. And so what we want to do, instead of building a shell, we want to build a nanowire. And so this is actually a virus with these 2,700 copies of these proteins on the end that we're now going to have modified through our library. And we are searching through our library for one that can build structure materials. And so these are actually semiconductor materials and nanowires that are made out of electronic materials. And so this first example that we're using here is a virus that we pulled out that can grow gold nanowires. And so through this selection process, we found one that was really good at grabbing gold ions out of solution and building gold nanowires. So this is not alchemy. You're not taking one element and making a different element out of it, but you're actually building a template to structure it in a way that you have it's over 99% efficiency in grabbing these ions out of solution and building the structure you want to build. So in this project, we've made over 110 new biotemplated materials. These materials over here are used for energy storage, catalysis, magnetic materials. We have miscellaneous materials, solar materials, and the interfaces between these. And so they're all very focused on making materials that have an impact. And so these are these are transmission electron microscope images. The scale bar here are on the order of 15 nanometers in size. So these are tiny wires that are gold. And so with different sequences, different chemical processing, we can control the diameter and length of these nanowires. And then we can actually start growing all kinds of different kinds of metal nanowires. So this is a gold, platinum, palladium nanowire, gold, platinum nanowires. We use many different kinds of combinations of these, and we've used it mostly for catalysis. This particular example that I'm showing here is a nanowire. We have a virus that can grow these gold nanowires, grows these nice gold nanowires seen in the figure B and C. Figure B. Figure C is actually an elemental map using high resolution transmission electron microscopy. We can map out the elements on this, and this one is showing the green is gold, and the red is platinum. So we had a virus that could first grow gold and then grow platinum on top of that in a really controlled way. And then we wanted to use it for a fuel cell. Let's see if I can move this away. I don't know if you're seeing this part of the video or not. So we could use it for a fuel cell. In this case, we were using it for a direct ethanol fuel cell, and we were able to use biology to grow both two electrodes, a negative and a positive electrode for an application in a fuel cell. I'm not going to go into too much detail of that, but basically what we're able to do is start to go in and change the sequence and change the chemical processing so that we could make a more and more efficient and high activity fuel cell. And so in this reaction here, the blue is the control. The blue is a platinum carbon electrode, and the black, the orange, and the green are going in and using biology to make different combinations and ratios of gold to platinum for two different kinds of reactions. One is for the ethanol oxidation, so it's for both parts of our fuel cell. And we showed in one case it was 2.8 times higher than platinum, and the other was 8 times higher than platinum. And so we were showing that something made biologically and through the process of evolution and selection could become better than the control, what was used today. So a lot of what we've done in my lab over the last couple of years is how do you go from a DNA sequence to a device. So you want to be able to just have a DNA sequence and go all the way up to a self-assembled working device that is cleaner and that in many cases are as good or better than state-of-the-art for applications. And one of the first applications that we worked in my lab were batteries and energy storage, lithium-ion batteries. And we've worked in many different kinds of biological-based lithium-ion batteries, anywhere from little micro batteries to ones that are used with drones and UAVs, to ones that are integrated into textiles, to vehicle batteries for lithium oxygen ones. And my students always wanted to work on grid or microgrid applications. And I always said, oh, we're never going to get there. Let's focus, on this. And just in the last year or two in my lab, we've made some breakthroughs to think about how to go to grid and microgrid storage using biological control of energy storage applications. And so this particular example is for lithium-ion batteries. And you may remember that in the lithium-ion batteries, we have a negative electrode that's an anode material, a positive electrode that's a cathode material. We have a separator, and we have the lithium ions. So the first thing that I'm going to talk about is the use of the anode or the negative electrode material, and the second part I'm going to talk about is the cathode material. So we'll start with the anode materials. And so what I'm looking to do is use biology to replace what's currently used. Some of this data is from our very first experiments many years ago, and our ideas have changed a lot, but I'm going to take you a little bit through the progression of this. And this is basically showing you're storing the lithium ions in one of the electrodes or the other. So the basic idea here was that we were going to use biology as a building block to template these materials. by giving them the right sequence to grow nanowires. These nanowires would then self-assemble themselves into a battery electrode material. What I like to say is that when you go along in this process, if you find a protein sequence that that can bind but it's not perfect, you have evolution on your side to try to make it better and better. And that's what we do. Basically, we went through that selection process I talked about. In this case, we chose cobalt oxide as our starting material. I'm no longer a fan of cobalt because of its impact on the environment, but this is where we started several years ago. Then you can go in and give it multiple genes for multiple possibilities, and so I'll take you through a little bit of that. So what we did was we grew these electrodes in the lab. This particular picture here is a transmission electron microscopy image of a virus that is nucleated, very tiny. These are less than 4 nanometers, so the size I showed in the beginning schematic of cobalt oxide, all grown in basically a shaker in the lab. And what we're going to be looking at is the specific capacity versus the cycle. So this is the ability to store lithium in the lithium-ion batteries. And so you want to have a higher capacity, and you want to have, obviously, a long cycle life. We had very short cycle lives when we first started this, but basically, we find the right virus sequence, we get it to grow the material of interest, we spin it down, we make a electrode out of the material, and then we measure it. And so we're looking at specific capacity versus cycle, and we're looking for this for an anode material and negative electrode material at first. And this is what we had. This is basic copper, and you have your cobalt virus, and you have your separator of an electrolyte, and you have lithium you're using just as a counter electrode for testing. So here's the commercial specific capacity here in black. Here's the theoretical capacity, and you can see that these diamonds and squares are our real data, that we actually hit the theoretical capacity for this, and then it starts to fade as you start to cycle. So we're happy, and then less happy, less happy, less happy. But we showed that it actually could work. And in this graph over here, we're looking at our virus templated nanowire versus just precipitating cobalt oxide out of solution, and it's not very effective at all. So we actually showed that it was about 2 times higher than a carbon-based anode, and carbon-based anodes are common anodes used today. But then my students were very clever, and they go, okay, well, we only used 1 gene. What if we go in and give it a second gene? We know that we'd like to have a good ionic conductor, which in this case is going to be our cobalt oxide, and we want to have a good electrical conductor at the same time. What if we went back and gave it one of those sequences we know are good at growing gold, so we could grow both materials simultaneously? And that's what they're able to do. Now we're looking at capacity again versus cycle, and this is our 1 gene battery here for just cobalt oxide, and now we have cobalt oxide and gold, and we get a higher capacity, we get better cycling and a faster reaction time. And so it was all based on being able to control DNA sequences that code for these different protein sequences, where these protein sequences could simultaneously grow 2 different materials, 1 good ionic conductor, 1 good electrical conductor. And so we are pretty happy about that, and we were funded by the Army to work on this. I went to present this in the Army, and they said, you only made half a battery. Where's the other half of your battery? They wanted to know where the cathode material was. And I went back to my students. These are 2 students, Yoongjung and Kyungjung here, and I said, you only made half a battery. Where's the other half? And they said, well, cathode material is actually more difficult. And we sat about thinking about how to do that. And so what we decided to do was to now go in and genetically engineer multiple parts of the virus. We decided to take this tip part here and engineer it to bind what's called a single-walled carbon nanotubers. Those nanotubes I told you at the very beginning, they're about 1 nanometer in diameter. They can be really good conducting materials. And so my students went and through that process of selection, give your viruses the ability to work with carbon nanotubes until you find one that's really, really good, isolate it out and amplify it. Now they can bind these carbon nanotubes. They said, okay, now so that's our good electrical conductor. Now let's give the ability to grow a good ionic conductor, iron phosphate material all along the outside. And then let's see if we can have it self-assemble into the battery. And it worked. This is a picture of a single-walled carbon nanotube, which is basically graphene, which is atomic level carbon that is, in this case, rolled into a tube, a sheet that's rolled into a tube. You don't actually roll it, it's grown into a tube. but they have all kinds of really, really interesting properties, but they're a nanometer in size. They're absolutely teeny. So how are you going to pick them up? Well, you engineer a virus to pick them up, grow another material on top of it, and then move it where you want it to be able to go. So this is a picture of what that looks like a little bit. These are transmission electron microscopy images. The higher contrast, the darker images are the iron phosphate. The tips, the wispy materials going by are actually carbon. And so it's this kind of beautiful structure that acts as a superhighway to get electrons in and out very quickly. It was very exciting. This is the idea of a superhighway. There's basically, they are connected to a current collector, and the electrons can be transported very, very quickly, very efficiently along these single-wall carbon nanotube highways into these electrodes, in a short amount of time. And so it actually worked out pretty well. And this is data looking at it. This is specific energy versus specific power. And the green is our 1-gene battery, 2 is our 2-gene battery, and blue is our best 2-gene battery. And so you can see how it's becoming better as a function of time. And if you look at it here, you're also seeing we're getting better cyclability. So now you're looking at 8 cycles, 10 cycles, you're looking at 50 cycles, which looks really good compared to our 8 cycles. But realistically, you don't want to have a battery that only cycles 50 times, and then you have to throw it out. So we needed to work towards 1,000 cycles, of which we've done quite a bit since then. Well, this battery was a record-breaking battery published in Science, and it went to a press release at the White House. The battery was invited to the White House. I wasn't invited to the White House, but I got to watch it on TV. My students, I watched it on TV, but it went for a press release on clean energy because it was a genetically controlled, biologically made battery that had excellent performance, and it was made environmentally friendly. Now you can actually go and you can make these into thin films, and you can integrate them into wings of UAVs, which has been a big project in my lab, trying to increase the amount of time that you can flight for UAVs, which are unmanned vehicles or drones. So increasing flight time for those. And so we were pretty happy in lithium-ion batteries and working towards that. And what my students always do is they always push me farther and farther. And they said, you know, lithium-ion batteries are pretty good. Here's a review article on specific energy versus different batteries and what the technologies are being developed at the time. And what you can see is where lithium-ion batteries are, and these are basically distances between charging, and we really wanted to be out here, 400, 550 kilometers or so. And so my students said we really need to work on lithium air batteries or lithium oxygen batteries. And so why don't they work that well right now? There's a large overpotential, low current density, cycle life, and cost efficiency. And so we decided to see if we could solve any of those problems with biology. And so my students went and genetically engineered viruses that were really good at growing manganese oxide, the really spiky manganese oxide material, which is a catalytic material. And I'll show you a video of what that looks like. But they started engineering kind of complex materials that could have catalysts associated with them, and these manganese oxide materials at the same time, and started testing them as lithium oxygen batteries. So here's a diagram of it. You have a virus, it's in water, you grow manganese oxide, and you give a little palladium. And this is a reaction, the reaction's going really quickly. But the idea is that it's gonna be much lighter weight because you're gonna be using oxygen from the environment as part of the reactant for your battery electrode. I wonder if I can back that up at all. Okay, here, I'm going to make it go slower since I can control it. So here we go, back again, just in case you're interested in this. You have manganese oxide, you have palladium, you have lithium plus oxygen and 2 electrons. This lithium peroxide, which is the discharge product. And one of the problems with these kinds of batteries is that we would get stuck in the discharge product and make it non-reversible. And so our material was much better at making a reversible reaction. So here's the reversible reaction here, where you have the lithium peroxide plus 2 electrons plus lithium going back to oxygen again. And you need this to be a reversible process. And so we were able to do that by structuring the material, giving it this spiky structure, the combination of materials. And the idea was to eventually be able to increase the distance between charging. And so we did that by making these two components mixed together. We had 45% improvement in the specific capacity, 7 times higher current density. And importantly, what we did was decrease the amount of noble metal, because obviously you're not going to want to have a lot of expensive metals in your battery electrode material. And so this graph over here is looking at specific capacity versus different ways that we synthesized it. If you look at the manganese oxide materials by themselves versus templated biologically versus doing them both with the palladium and the biotemplated manganese oxide, we got the best materials. And it was state of the art at the time we published it for these types of materials. And then we went off and explored many different kinds of materials using the same kinds of technologies. This is kind of a fun experiment. This is, we used a virus to make an aerogel, a super lightweight battery that had a very, very high porosity, and the battery was sitting on top of a flower, very lightweight, high energy density material. But then what my students said was that that they wanted to be able to get away from any toxic materials at all, any cobalt materials. They wanted to think about how to get away from lithium because lithium was in short supply. So we started thinking about more earth-abundant materials. And so this is a very long story, but basically we started engineering these materials with different kinds of metal oxide materials that were sodium, nickel, manganese, titanium oxide or tin oxide or silica based materials to be able to make sodium ion batteries better batteries. And part of that was the idea to get away from lithium as a get away from lithium batteries to sodium batteries because lithium is more rare. It's not actually mined in the United States. It's obviously very abundant in the ocean. If you could mine it from the ocean, a lot of people are doing that. But we also wanted to get rid of cobalt as well. And what my students wanted to do was to move away from cobalt-based materials because of the how bad cobalt is for the environment and all the cobalt poisoning that comes with mining materials for batteries as well. So we focus a lot on getting away from those kind of materials. And so there's a lot of pros to sodium batteries, which they have similar electrochemistry compared to lithium ion batteries. It's cheap and abundant. And you can think about changing the current collector. You can have aluminum instead of copper, but there's a lot of cons as well, which is the redox potential is above that of lithium. The sodium ion is larger and less polarizing, and there's diffusion effects. And so my students using genetics, so this is, I'm seeing this again, learned how to engineer around that and make these new combinations of materials that hadn't been able to be made at the nanoscale before. And what they did was actually make very high energy density materials that have very high cycle life. So some of the capacities surpass current lithium-ion batteries with cycle lives above 1,000. We were looking at other kinds of batteries as well, and let's see, I'm going to see if I can put it on a pause here and come back.